Hello, today's topic is prospects of genetic engineering in the treatment of cervical cancer. Dear participants of the 47th World Seminar on Women's Health and Nursing SciTech Central Inc. Let me present you with a review of the literature in international networks dedicated to the treatment of cervical cancer caused by the human papillomavirus. Cervical diseases account for 12.2 to 22.5% of gynecological diseases and occur at any age. Due to the risk of developing a malignant process, timely diagnosis, adequate therapy, and rehabilitation for prevention are required. Since cervical cancer is the third most common malignant neoplasm among gynecological oncological diseases in women around the world, according to the World Health Organization, in 2020, more than 500,000 women were diagnosed with cervical cancer, and 342,000 women died from this disease. Without additional action to control cervical cancer, the number of new cases per year between 2018 and 2030 is expected to increase from 570,000 to 700,000, and the number of deaths per year is expected to increase from 311,000 to 400,000 women. Currently, a cause and effect relationship between the human papillomavirus and tumor transformation of the cervical epithelium has been proved. Today, there are more than 224 types of human papillomavirus. Of these, the anogenital area is affected by more than 30 types, 15 of which are oncogenic. Human papillomavirus 16 and human papillomavirus 18 have the greatest oncogenic potential. Then, in descending order of frequency, 45, 31, 33, 52, 58, and 35 types. The 27 human types of human papillomavirus are divided into three categories. High risk, 16, 18, 31, 45. Average, 33, 35, 39, 51, 52, 56, 58, 68, 73, 82, 83. And low risk, 6, 11, 26, 40, 42, 53, 54, 55, 57, 66, 84. Human papillomavirus is an obligate intracellular parasite that is usually present in the episomal form and is transmitted to a sexual partner. The malignant process progresses through the stage of cervical intraepithelial neoplasia, SIN, with the possible development of the progress against the background of benign and tumor-like diseases of the cervix. After penetration into the epithelial cell through the basement membrane, when microdamages occur in the transformation zone of the cervix, viral DNA penetrates into the nucleus of the host cells, integration, and changes the cellular DNA replication program, causing a malignant transformation, neoplasia. At the same time, the virus reproduces new variants with stimulation of proliferative activity under the influence of proteins E6 and E7, with the release of proteins P16INK4-alpha and Ki-67, which are prognostically significant for the development of neoplasia. Genetic destabilization during inhibition of tumor suppressor mechanisms lead to the emergence of mutations that ensure cellular immortality, and oncotransformation, which characterizes the model of carcinogenesis. The heterogeneity of cytopathological changes, the absence of viremia, inflammation, a decrease in the severity of innate immune response, immune suppression, T lymphocyte anergy, and the absence of immunological memory contributes to the long-term persistence of human papillomavirus. Long-term persistence and activation of the human papillomavirus can activate the complement system, with the occurrence of endothelial dysfunction and contribute to the development of autoimmune disorders. The co-founding HPV-associated neoplasias are genetically determined anaerobic dysbiosis of vaginal comedones, which develop against the background of L. iners. Predictors of neoplasia are also hormonally associated gynecological diseases with the dominance of the estrogen metabolite, 16-OH hydroxyestrogen, Immunosuppression and psychological maladjustment, a feature of viral invasion, is its chronic form, characterized by periodic periods of exacerbation and elimination, ensuring lifelong persistence of the virus in the human body. Despite antiviral therapy, many drugs do not have high specificity and effectiveness, which is associated with the high adaptive ability of viruses to treat. All this requires in the search for new effective methods of therapy. It is worth noting the responsive adaptive ability of viruses to the applied treatment methods. Despite the improvement of invasive properties of drugs to overcome biological barriers, the development of the disease has a long course, often leading to death of the recipient, which is the development of malignant disease of cervical cancer. It is known that different types of viruses have different ways of assessing organs and tissues, and the mechanism of transmission of viral infections differs from other microorganisms, in that at the initial stage they persist in the periphery, and only at late stage does the possibility arise for the development of a malignant process. All this requires the development of more effective treatment methods, one of which could be the technology of genetic scissors. The development of modern technologies for editing the human genome has made it possible, using genetic engineering to open up great prospects in the treatment of a number of diseases that were previously incurable. CRISPR structures, cluster regularly interspaced short palindromic repeats, were first discovered in the genome of Escherichia coli in 1987. Years later, it became known that these sequences can accumulate as a result of invasion of mobile genetic elements, MGE, mobile genome express, bacteriophages, plasmids, and transposomes, into a prokaryotic cell, which form the basis of adaptive and inherited immunity bacteria, archaea. Emmanuel Carpenter and Jennifer Doudna received the Nobel Prize in Chemistry for the development of a genome editing method. This is a promising genome editing technology, but there are a number of limitations and challenges that need to be addressed before its clinical application. One of the biggest obstacles is delivery efficiency, since CRISPR-Cas9 assumes an intracellular mechanism of action. In this, the components can be delivered in various forms: as small, conveniently packaged mRNA or as plasmid DNA, encoding the Cas9 protein. Other advantages of mRNA delivery include highly active gene editing and control over the volume of delivery into cells. Plasmids have limitation in size and off-target effects, but also have the advantage of stability and flexibility in design. Viral and non-viral methods of material delivery, such as electroporation, microinjection, and lipid nanoparticles, exist, but their therapeutic applications are limited due to their relatively low delivery efficiency. The purpose of the article was to analyze modern methods of treating human papillomavirus. Materials and methods, a review of publications in international search engines, was carried out using keywords. An alternative treatment could be CRISPR-Cas9 genetic scissors technology for editing the human genome, which makes it possible to remove structural elements of viruses after identifying a foreign agent. Of interest are human papillomavirus, which are small, non-enveloped dsDNA viruses. Highly oncogenic strains of human papillomavirus are the cause of rectal cancer in 95% of cases, oropharyngeal cancer in 70%, and vaginal cancer in 60%, and are the leading cause of cervical cancer, which is associated with high mortality. Current vaccines prevent infections with the virus, but do not protect those who are infected. All this significantly limits the application, since the spectrum of human papillomavirus genotypes does not have a cross effect. The use of CRISPR-Cas9 in combination with a modern anti-cancer drug can become an effective method for treating oncology, especially in cases associated with HR, human papillomavirus. Experiments with intratumoral administration of CRISPR-Cas9 mediated by human papillomavirus E6 and E7 led to the development of an inactivating mutation, CAS9 indel, which is associated with the induction of p53 or PRB proteins and can lead to cell cycle arrest and cell death. Today, the CRISPR-Cas9 system has an enormous potential for use in human genetic engineering, both in vivo and in vitro. But there are certain questions associated with the introduction of this technology into clinical practice. One of them is the off-target effect of the system, which causes the occurrence of side mutations. Another is the Cas9 expression level target sequence and quantification methods that determine the cleavage rate of non-target level of CAS9. Future research should focus on developing a new, robust, and more targeted method to improve the specificity and targeting of CRISPR-Cas9 technology. For the successful application of the CRISPR-Cas system in the gene therapy, a new research strategy must also focus on improving the frequency and efficiency of site-specific nuclease, especially in genome editing. An important role in the successful application of this technology belongs to the delivery of CRISPR-Cas9 components to the target cells. Since the delivery system used is not specific and highly effective, which is especially important in the field of biosafety. Another problem for scientists in NHEJ, which leads to the generation of stable mutant viruses that can resist Cas9 sgRNA, while causing DNA repair in host cells. Such limitations in the use of genome editing technology open up new opportunities for research. So, the use of CRISPR-Cas9 in clinical practice requires significant improvement of the method, which could result in the development of a promising direction for the treatment of a wide range of diseases. Thus, the use of CRISPR-Cas9 targeting E6 and E7 oncogenes of human papillomavirus 16 in combination with cisplatin in vitro and in vivo can serve as an effective therapy for cervical cancer, which will reduce mortality from the malignant disease. The technology of genetic scissors, the development of modern technologies for editing the human genome, has made it possible, using genetic engineering to open up great prospects in the treatment of a number of diseases that were previously incurable. Thank you very much for your attention. I hope you enjoyed the presentation. Bye-bye.